So, to put some context around Germinate, we have this challenge, and everybody will be very familiar with the challenges that we have now with scientists producing much larger data sets. Generally, they're much more complex as well. And that mixture of complex and large is what people call big data. So we need to find ways that we can, an infrastructure we can put into place to allow us to do things with that data. So we want to be able to store it, explore it, interact with it, and try and understand what it means. But to do that, we need to develop tools that help provide insight into this complex data. And that's kind of where Germinate comes in. There are, of course, lots of other tools as well, but today we're going to focus on Germinate. And we need to provide the data to people in easy to access chunks. So it's no use giving somebody millions of data points and saying just go and do something with it. So what we want to try and do is give you ways to explore that data and find the bits that you're particularly interested in. And that might allow us to see relationships and things between data that may not be visible if you're looking at it, for example, in a spreadsheet. And I think what's really important is it's not about telling you what to think. So you guys are the experts within your areas, and it's our job to give you data, but it's part of your job as to how you interpret that data. So it's about giving you tools to look for patterns or visual cues that may point you to results that are interesting. So why are we doing this? Well, very quickly, the main reason for doing this is we want to make experimental data available to people. So in our area, we're looking at things like pre-breeding. So we basically want to make that diversity available to people in ways that they can use it and exploit that to do something cool down the line, and hopefully create some potentially new crops that are more adapted to the climate or pests and diseases, that kind of thing. So we're going to show a video here. This is in a couple of minutes, just a couple of minutes about introduction to Germinate before we start. So let me know if you can't hear this.
Germinate is an open source plant information platform that's used to store experimental data from plant genetic resources collections. Why use Germinate? Germinate allows you to manage data from your collections in a standard and fair way. It allows you to explore and make your data available to collaborators via intuitive web-based interfaces. The single point of access for all background data on genetic resources collections that Germinate implements provides a valuable digital curation resource, as well as offering essential continuity on how new data is recorded, archived, and made available to collaborators and communities. What types of data does Germinate store? Data types include passport, phenotypic, field trial, pedigree, genetic, climatic, and geographic location data, as well as user-submitted annotations and images. Where is Germinate developed? Germinate has been developed by the Information and Computational Sciences Department at the James Hudson Institute, just outside of Dundee in Scotland. Originally, Germinate was created to meet the needs of barley and potato work being carried out in Scotland. How long has Germinate been in development? Germinate was originally the subject of a publication in 2005, but it has advanced considerably since then. From originally only storing a few markers and germplasm entries, we now have databases with hundreds of thousands of plant lines and millions of SNP markers and phenotype data points. What species use Germinate? We have a wide and varied list of crops that use Germinate, everything from barley to sweet cherry. Does Germinate support standards? Germinate is fully compliant with the multi-crop passport descriptors developed by the FAO, and we are developing support for data initiatives such as MyAPI and Dublin Core. Germinate aims and supports making data available using FAIR principles, and we are working to include DOI features and tools for datasets and germplasm. Once my data is in Germinate, how do I get it out? Germinate offers different formats to download your data, from plain text formats to formats specific for external analysis programs. One of our main underlying beliefs is that any data you put in, you can get back out again quickly, and there are tools to give you an overview of the data that your database contains. Have a play about with our demo database to get a better understanding of the tools Germinate provides, and don't worry, you can't break it. Can I install Germinate myself? Of course, you can download Germinate from our GitHub repository or using a handy Docker image to get you up and running quickly. You can even use your Raspberry Pi. If you want to just have a quick play about with the interface, you can use one of the publicly available databases or our demo database, which has an example of all data types. That's great. How can I get more information? Contact us. Please get in touch and we can give you an extended demo or talk you through using Germinate with your own data or how you can help be part of the Germinate community. Germinate continues to see long-term development and stability through core support from the James Hutton Institute and Scottish Government, as well as funding from a number of organisations and national and international funding bodies, such as the Crop Trust and the European Union. Go on, give it a go and get in touch.
Oops. I'm sure we don't want to watch that again just now.
Sure.
Okay, so that's a quick introduction. I've just got one more slide. So just to put things into context as well. So people often ask, like, where does Germinate sit within the GLIS, Global Information System from the FAO? And just to give you an idea of some other tools that are around about that do similar things. So this is a graphic from a book that's coming out next year that we're involved with, along with some guys from IPK and the Crop Trust. And we broke down information systems into 3 different pillars. So there's at the top here, there's germplasm collection management. That's the kind of things that you're doing at your gene bank level. And we've then got the germplasm gateways. So in Europe, that's Eurisco, and then the worldwide system is Genesis. And then we have tools which sit on top of these, which provide a much deeper set of data. And that includes things like Germinate, Germinate, that kind of thing. So that's where we come in. So that might give you an idea of where Germinate sits within other information systems that exist. I should add that we also do stuff on gene banks as well. So while Germinate is down at the bottom here under C, we are in other areas too, but just not quite as prominently. So I'm going to shut up now. I'm going to pass over to Sebastian, who's going to give you and show you some of the features and stuff you can do with Germinate. So as I said before, please, any questions, just jump in and ask, and let's keep this interactive. Okay, Sebastian.
Right. Let me know if you can see that or not.
Yeah, it's
right. So most of what I'm going to be doing today is going to be like an interactive demonstration of Germinate. I've just got one slide at the very start where I just wanted to highlight, the different things that I'll be talking about today. So first off, I'm going to start with an introduction, just explain the main interface of Germinate, explain where everything is and how you would find, the different types of data. After that, I'll give you an overview of some overview statistics with the finger millet and grass pea database being the examples here. Then I'm going to look at germplasm data because everything in Germinate kind of evolves around the germplasm. So I'm going to show you how you look at germplasm data, how you filter it, and how you search for specific entries of interest. After that, we're going to look at some trials data. And I will be using some examples from the finger millet and grass pea again. But I will also show some recent development here with where we've incorporated drone imagery, which is barley, based on barley data. Then we're going to have a quick look at genotypic data. Now, because the output that comes out of Germinate will, by default, or one of the options is to open it up in Flapjack, and we've got a separate Flapjack training coming up in, I think, a few weeks' time. I'm not going to go into too much detail there because, I don't want to take away anything from the other training course. And then finally, there will be a quick look at some geographic data as well, how you make queries based on, points on a map or areas on a map if you're interested in specific regions. Okay, so let me switch to the demonstration part then. So this is what Germinate looks like, and I've got 4 different instances of Germinate open here. I will be switching between them, but I will always explain which one we're looking at just now so that people don't get confused. But if I switch between them just now, you can see that they've all got a very similar layout. So in all cases, the main navigation menu is along the side. It takes you to all the different sub pages of Germinate. So under the data heading, for example, is where you find all the different data types that Paul mentioned. So you've got the passport data, genotypic data, trials or phenotypic data, and then also some geographic or climate data here. Below that, there's a section on optionally you can link publications to Germinate, and you can link it to specific entries in the database as well. So, for example, if you if you need to make a data set available for a publication and you're hosting that in Germinate, you can link to that publication from here as well. Along the top then is the other part of the navigation. This Germinate logo up here will always take you back to the front page. So if you're if you navigate through all the data pages and you just want to get back to the front page, you can just click on that here. The search box up here is a search that searches everything in the database. So if you're ever looking for data related to a specific germplasm, or if you're looking for a trait or a location or anything really, you can just type that in here and use the search functionality to find it. And I will show an example of that in a bit. The next button that is next to this here is this question mark. Some pages will enable that, and if you click on it, it gives you some more information about the page that you're looking at just now. So it's just more help information if you're not quite sure what you're looking at. In some cases, there will be other buttons with question marks on them, dotted around the interface. In this case, if you click on this one, it will explain how the table works and how to use all the individual functionalities that are available within this table. Now, continuing up here, the next button over is just to switch between a light and a dark mode, because sometimes you're working in the dark and it's a bit easier on the eyes if you just switch to the dark mode. In some instances of Germinate, we provide translations of the user interface. So in this case, in our demo database, we provide this in English and German. And if that's available, you can just easily switch between those, and you can see how the text on the interface changes based on the selection that I've made. The last one up here that I want to explain is this, what's called a hamburger menu because of the three horizontal bars. If you click on that, it's gonna expand the asynchronous job sidebar, which basically is the place where if you're uploading or downloading data, this is where your job will show up because it runs asynchronously in the background. So you can just close this, continue browsing the website, and at a later point you can come back and then your data that you exported will be available here. Right, so let's switch over to Graspy and let's look at some overview statistics. So under the data menu, there's the statistics page at the very bottom. And if we click on that, we get a very broad overview over the data that is in this database. So first of all, we've got what's called a sunburst diagram of the taxonomy. So sunburst is kind of like a pie chart, but with different levels. So you can see that at the center, the genus, then the next ring over is the species, and then if available, the subspecies further to the outside. This is fully interactive, so you can hover over it to see the number of germplasm for each of the taxonomies. You can click on these to have a closer look at the subtaxonomies. But also if you select, for example, this one here, it will take you to the germplasm page, which is now pre-filtered with the selected genus and species. So you can, if you're looking for a specific taxonomy, this is quite an easy way of just finding it by clicking on it in the sunburst diagram. The next overview statistic to the side here is breaking the germplasm down by its biological status. And in this case, we've got a pretty even breakdown between traditional cultivars and wild varieties. They're about 2,500 each. And then there are a few advanced or improved cultivars here as well. Similarly, again, you can click on these to see the germplasm that falls into that category. And below that is what's called the Passport Data Completeness Index. Now, this was published by Theo van Hintem in 2011, and the link to the paper is here. But it's basically a score for each germplasm that tells you how complete the passport data is. It's a value between 0 and 10, 10 being a full set of passport data. So the further to the right these bars are, the more complete the database is. In this case, most of the germplasm has a passport data completeness index of between 5 and 7, which is pretty good because we've seen other collections which are more skewed towards the left side of this graph. Below that, the last thing on the statistics page is a geographic overview. In this case, we've got a heat mapped view of the world where you can see how many germplasm are from which region. Now, the darkest one here is Bangladesh, because more than 1,000 of the germplasm are from that region. Then there are places like Syria, Turkey, and Morocco, which are still quite dark. Also Ethiopia. And again, you can click on these to see the germplasm from those regions. Now, if I go back to the germplasm page, this is probably the page that most people will look at first because it shows you all the germplasm that is in this database. In this case, there are about 5,600 individual entries. And this table shows you a subset of what's called the multi-crop passport descriptors. So it shows you things like different identifiers for each germplasm. There are DOIs here as well for those where it's available. Then we see the biological status that we saw in the bar chart earlier, the taxonomy. Then there are things like the location where it has been collected. So this one's from Cyprus. And if you click on it, you can actually look at the map and zoom in to see where in Cyprus this was collected. And then at the end, we've got the passport data completeness index again, just for reference. Now, all of the tables you see in Germinate have got functionalities added to them that make them a bit more useful rather than just looking at the data. So first of all, there are the columns that you see, you can change which ones are visible. If you're not interested in, for example, the synonyms or the collector number, you can just hide those. Or if you wanted to make, I don't know, the latitude and longitude visible as well, you can do that here and they will become visible in this table. Something else that we can do is obviously you can sort if you just click on the column header, but probably more interestingly, you can filter this table by clicking on this funnel icon up here. And that will allow you to specify the column that you want to search, the operator that you want to use. So if you want what you're searching for to be equal, between, so if you're looking for something in a range, if you're looking for something that's just greater than a specific value, you can provide that here. Additionally, we define some common table filters above. So these are things that we think most people will be filtering on as a first instance, and then you can adjust the filtering later on. So these include things like whether the germplasm has data for a specific type. So if I click this button here, that will filter the table to only include germplasm for which there is trials data. That has filtered it down to only 500 records now. And I can also select a country, for example. So let's pick where is it? Let's pick Syria here. That narrows it down to 115. And then let's say I would also only want germplasm from a specific elevation. And now that I can adjust by, again, clicking on the funnel icon here and using this add button. So I've seen these two conditions up here are the ones that I selected previously. So it has to have trials data and it has to be from the country Syrian Arab Republic. And if I want to add another condition to this, I can do that by simply clicking on the add button, then selecting what it is that I want to filter on. So say elevation should be greater than or equals, let's say 500. And if I adjust that, then it'll only return the ones with an elevation above 500, and there are only 34 of these. Now, filtering is all well and good. It shows you the germplasm that matches the criteria that I selected. But what do we do with them then? And that's where the last column in this table comes in. This is where you can mark specific entries, which adds them to the list of interest. And that's your list. It's specific to yourselves. So you can kind of imagine this being like your online shopping basket or something. You add items to it, you can remove them again. and it will just keep track of the material that you're looking at, which becomes useful later on when we're looking at data visualizations, because then you can highlight that list and compare it to the rest of the database. Now, rather than selecting them individually, I can use this button here and just say mark all, which will add all of the 34 germplasm that match the criteria that I filtered on to my list, which has now been updated to 34 entries. Right, let's quickly switch to finger millet and also look at the passport page. Now, in this case, let's say I want to also filter for germplasm with trials data. And then I want to look at the one that has the most complete set of passport data. So if I filter on the passport data I sort by the passport data completeness index. This one here has a score of about 7, and it's the highest one. So let's look at this one specifically. So if you click on any of these identifiers on the left, to the left of the table, in the left of the table, you get to what's called the passport page. And that's a page that shows you all the information we have about the specific germplasm. So again, you see things like the passport data completeness index. You can see things like the multi-crop passport descriptors, which is the FAO standard for describing plant material. You can see different synonyms for this germplasm. But if I scroll down, there are other things of interest, like, for example, the data that we have for this germplasm. In this case, there is genotypic data and there's phenotypic data. And you can see roughly the number of data points in each of these data sets. What's also interesting here is the license. So all of these, because they are from the Crop Out Relatives project, they have what's called a Creative Commons license associated with them. And that just means that people have to accept these license terms before they can use the data. And just by clicking on the license, you can read through this. And before you can use the dataset, you actually have to accept these terms. Now, what that does then is, once I've accepted these terms, I can look at the germplasm trait performance for this particular germplasm. And that basically shows you for each of the traits that it has been that have been scored for this germplasm. It shows you, where does it sit compared to the rest of the database. So for example, the neck blast severity, it's got a very low score for that. It sits at the very left of this range. Whereas days to maturing, it sits like at 2/3. So it's later in the season than other varieties or other germplasm. And things like the threshing percentage, it sits somewhere in the middle. There are other sections on this passport page, like pedigree information and images. The two databases that I'm showing today don't really have any images, so I'm going to skip that for now. But let's now have a look at the search. So I said you can paste anything into the search. So let's just paste Karakana in here and search for that, and that will return, the data broken down into the different data types that are available. And in this case, it tells us that there are just over 1,000 matches within the germplasm data. And that's not surprising because it's the species term, and it just filters everything down to all the ones that match the specific search term that I've searched for. So that was part of the taxonomy that I was looking for. And let's say I'm interested in anything to do with BLAST. And I'm going to change the comparison method here to contains. So it'll look for BLAST anywhere in the database entries. So if I do that, that changes the results. And now I see things like germplasm attributes. So for this germplasm, there is a remark that someone left that just says, it's resistant to pests and moderately resistant to neck blast. So that's where the match blast comes in. There are about 20,000 phenotypic data point matches here for BLAST. And these are both based on the trait name BLAST, but also on the data set description, which also contains the word BLAST. So it'll return everything, that matches the search term that you were looking for. Additionally, there are 5 data sets that reference the term BLAST. So there are 2 genotypic data sets and 3 trials data sets here as well. Which brings me to the next section that I want to look at. So if I switch back to GraphP, let's have a look at some trials data. In this case, there are 8 different data sets, and you can see the license terms. I have not accepted them on this database yet. So let's just do that. I click on here and I click on accept if I'm happy with what I've seen. And that, adds the green tick box and removes the red background. So now I can use the data within these data sets. So I'm interested in these 3 data sets down here. They are screening data sets for resistance from 2017, 2018, and 2019. And you can select them by ticking these checkboxes in the first column. And then once you're happy with your selection, you can click on the next button to then look at some data visualizations. Now, there are different options up here. The first one that's selected is like a data statistics page, where you can select different traits that are part of these 3 data sets that I selected. Let's just select them all. And then let's have a look at what that data looks like. So what this does is it generates box plots for each of the traits grouped by the data set that I selected. So these 3 first traits have only been scored in the latest, the 2018 data set. This trait here has been scored in 2 of these data sets. And if I scroll further down, the seed yield and the biological yield have been scored in all 3 data sets, and you can see the difference between the values in those 3 data sets. So it kind of gives you a nice overview over both what the data within the data set looks like, that's each box plot individually, but then also the comparison between the 3 different trials data sets here, so you can see how they vary across the years. If I then switch to the next tab up here, the interface is kind of similar. So again, you select some traits, and let's pick these ones here, and you can hit plot. And what this gives you is a scatter matrix of the different trait combinations. So each of these scatter plots here is a combination of 2 traits. This one up here, for example, shows days to flowering on the x-axis. On the y-axis, it shows biological yield per plot. These, again, they are completely interactive. You can hover over the data points to see the germplasm identifier. You can select different areas, which highlights the same data points in the other charts. But it also allows you to add those items to your marked list or remove them from your list. Other things you can do here are you can color these charts. Now let's color this first by our marked items, just to see how they compare to the rest of the database. So this will plot the 34 items on our list as green dots in this diagram, and the rest of the database are blue data points. So you can kind of see how it relates or how they compare to the rest of the data. Another coloring that we can utilize is the taxonomy, for example. So if we replot this colored by taxonomy, you can see how the data breaks down into the different taxonomies, which are shown in this legend down here. And particularly this plot is quite interesting because you can see that there's a grouping of these blue triangle data points down here, which are all from the same taxonomy. There are a few up here and there as well, but there's a big cluster of the same data points down here. And then things you could do is you could say, well, okay, I'm interested in these data points here. So you draw a rectangle around them, which tells you that there are 68 germplasm in this selection. And you could then add them or remove them to your marked list as well. So if you wanted to export the genotypic data for these lines, you can add them to your list, then head to the genotypic export page and export the data just for these selected items. Let's color it one last time, and this time by the data set. And remember, the data sets were from 3 different years. So you can see the purple one is 2019, green is 2018, and blue is 2017. And you can see how the data from the different years compares to each other. And again, you could now make selections here and add things to your list. Now let's move on to the next type of visualization that we have here, which is a bit more in detail. So for this one, again, you choose the traits that you're interested in. But this time you have to make a selection on the right as well. So in this case, we select the marked items, out of which there are the 34 that I filtered on initially. And if I then hit compare, what this does is for each trait, it will plot them side by side as a box plot. So this is biological yield per plot. And you can see the data distribution for each of the germplasm within that trait, which kind of gives you an idea of how they compare to the rest of the current selection. The dotted line in this case is the average across all the data, so you can kind of see if it's below or above the average. And as I said, this is done for each of the traits that I selected above. Okay, so much for the visualizations. It's always nice to look at your data in a visual way because humans are very good at spotting patterns in the data. But sometimes you just want to see your data in a table form, and we do this as well. So in this case, we see the germplasm, the data set, the trait, and then if I scroll to the right, we see the actual the trait value as well. And because it's in a table, and I said, the tables have got lots of functionality added onto them, I can filter this now as well. So say I'm only interested in the trait manganese, and I filter on that. That filters it down to about 300 data points. And now I could sort this based on the trait value, which gives me the lowest, or if I invert the sorting, gives me the highest scoring germplasm in the manganese trait with 95 milligrams per kilogram. That's this germplasm here. And I could now click on this to get to the passport page, or I could use this checkbox here to add it to my marked item list. Additionally, though, let's say I'm only interested in the ones that have a value between 50. So I add another filter and I say, okay, the trait value should actually be less than or equals, Jesus, put that there, less or equals 50, which then updates the table, and now the maximum score is 50. And again, I can mark this item if I want to, and this is the germplasm IG 64847, which is responsible for this data point. Now, let me show you some very recent development that we've undertaken for the trials data. And I'm switching to a barley data set now that we're currently working on. So this isn't public yet, but I'm going to give you a preview of it anyway. Where we've taken UAV or drone data and also time series data, just to look at how the different traits develop over time. So in that case, this time series tab becomes available. And again, we've got a selection of traits that we can select from here. So there are things like the NDVI or the digital surface map, which are things that are measured by from drone imagery. And if I plot this here now, what we get back is a visualization like this. So we can see that data has been recorded over a period of time. And the dark gray line shows you the average across the whole trial over time. And the light gray shows you the value of the range of values across the whole trial. So some of the germplasm will be sitting along the bottom edge, some will be along the top edge. It just shows you the whole range. And what it also does is, in this case, because we have information about the location of the individual plots, and we also have the imagery from the drone, we can actually step through the time points and see how the plot changed over time, how the trial and each individual plot changed over time. What this also allows us to do is make selections on the map. So I can click on each individual plot and I get the germplasm that's been grown in this plot. And what it also does is, if I select a few more, is it adds them to this chart up here. So I've selected 3 plots and they are kind of surrounded by these colored rectangles here. So there's like a purple, a green, a blue, dark blue, orange, and green. And you can see the same colors up here in this chart, and you can hover over it to see the the development for each of the germplasm over time. So there are a few that sit at the top edge, the three up here, and there are a few that sit more along the bottom edge. I can now change the trait that I'm looking at by just selecting a different one and hitting plot again, but it'll keep the selection that I've made. So it'll still show the 6 plots that we were looking at, again over time, so you can see the development. And the heat map on the map down here now reflects the other trait that I've selected. Interestingly, in this trial, is what we found is that this row here has values that are a lot lower than the rest of the trial. And that's the assumption is that that's because there's only one guard row on this side, whereas the trial is a lot more protected on all other 3 sides. So this kind of visualization is quite good at showing edge effects or patterns in the data, maybe because of, I don't know, flooding or something like that as well. Okay, let me switch back to finger millet now, and let's move on to genotypic data. Now, for genotypic data, we've got 3 different sections. You can look at the markers, you can look at the maps, and you can actually export the data as well. So let's start by looking at the map. Sorry? Okay, I thought there was a question, but I don't think that's the case. Right, so if I select the second map, for example, then I get all the markers with the chromosomes and their position. But what I also get is a density chart for each of the chromosomes or linkage groups. So I can see the density of markers on each of these. Rather than just look at it, though, you can also make selections here. So say you were interested in this region on chromosome 1b, this region on 3b, and I don't know, this region on 7b. You can then, similarly to the other visualizations that we've seen, you can mark the items in these areas, which then adds the 738 markers that are in this region to your list as well. So for example, if you use this to then export genotypic data just for those regions, it would only include the markers on your list. So let's move on to actually exporting the genotypic data. Let's pick this data set here. There are 3 available with the number of data points in them as well. This page then looks kind of similar to the trait visualization that we've seen, but instead of selecting traits and germplasm, in this case, we can make sub-selections on the germplasm and markers. So I could reduce the selection to the markers that I selected on the map chart, and also some groupings of germplasm that are available in Germinate. But let's just leave it on the whole data set. Then down here, I can define what formats I want to export the data in. So by default, we export to a tab delimited text file, but you can also export it to Flapjack, for which we will have a training course in the next couple of weeks, but also HapMap, just because it's a very, very common format for this type of data. Finally, if I then hit the export button, it will open the side menu that I mentioned at the very start, which shows your export data jobs. So this is the one that I just started. It's currently running, but I prepared this one earlier, so I can just download this now. So this has finished. And if I download that, it will provide a zip file that, if I open it, contains the file formats that I requested. So I wanted a Flapjack file, also a HapMap file, and these other 3 are plain text files that contain the genotypic data, the map, and also some additional identifiers for the germplasm. So then double-click the Flapjack file, and that's assuming I have Flapjack installed. That's going to open the data file that I just exported on the wrong screen. Give me a second. Drag that over here. So that'll open the data that I just exported from Germinate and Flapjack. Now, since we have the separate Flapjack training coming up, I'm not going to explain what Flapjack does or how it works, because someone else is going to cover that in a couple of weeks' time. So that's it for genotypic data. So let's have a look at GraphP again and look at some geographic information, which is the last section that I'm going to talk about today. So if we look at the locations in the GraphP database, this just gives us all of the different collecting sites with their GPS coordinates and the country they're part of. But it also shows them visually on a map. On the left, we see a clustered view, and on the right, we see a heat mapped view. And similar to all the other visualizations, this is fully interactive, so I can zoom in and I can select individual sites, and I could then click on this site to get all the germplasm that has been collected at this particular site. If I'm interested in searching for something rather than just looking at it on the map, what we can do is we can head to the geographic search page. A second here. All right. Not sure if my internet connection just went. Can you still hear me? Yeah, okay. Well, this will suddenly become unavailable. Oh, can you check at work if things are still online?
Yeah, give me two seconds.
Because it looks like one of our servers might have restarted, which is very unfortunate.
Yeah, it's down. It's down.
Right. In this case, I apologize. One of the servers at work at the Institute must have gone down. But I mean, we got to the last section that I was going to cover anyway. So I'll just explain briefly what it is that I wanted to show. So in the case of geographic search, we have got maps available where you can either pick a location on a map and it will return all the germplasm and collecting sites, that are closest to that point. But I can also make a shape selection. I can draw a polygon on a map and it will return all the locations or germplasm within the selection that I've made. So I'll keep an eye on whether this comes back online, but for now I will stop sharing and see if there are any questions in the meantime. Or did you see any questions
Yeah,
pop up?
so I apologize about. So just to let everyone know what's happened, it looks like all our scientific computing infrastructure has gone down at work, according to our monitoring tools. So I apologize for that. It couldn't have happened at a worst possible time than when you guys have all come along. But as Sebastian was saying, I think we're pretty much at the end of what you're talking about. So does anybody have any questions that they'd like to ask? So I could start, Sebastian, by asking when we're going to have the UAV and drone tools available for people to have a play about with.
Yeah, we're in the final steps of testing just now, so we're going to show it to some of the experts at the James Hutton Institute. We've started that already, and it all looks quite promising. All the upload infrastructure for uploading data of that type are in place, so I reckon it'll be very early 2023, so January probably, that it'll be available.
And I guess another question is, I'm not sure you might have covered it and I've missed it, but it's just talking about, so groups are really, really important and they're really powerful, but just talking about how you can share those groups with other people who you're working with, or maybe with the community in general, and the tools that we've got to do that.
Yeah. Yeah, I didn't cover that. So remember when I said you can add items to your list? So once you've built up a list that you think is useful to other people as well, you can persist that group into the database, which just means you create an entry in the database that is then made available to everyone else using Germinate. so that it becomes useful to those other people as well. And then they can use that same group that you created to highlight data points in the charts, to sub-select the data that gets exported to Flapjack or other tools. Yeah, it's just a very handy mechanism for making these sub-selections and sharing them with other people.
Any other questions from anybody?
Yeah, I have a question. Firstly, Paul and Sebastian, this is just incredibly amazing what you've done. It looks just amazing. So well done. The question I have is, I guess, two parts. Firstly, does it interact with programs such as Genesis, so that you have refined the list of sessions that you're interested in, or breeding lines you're interested in? If they're on Genesis, you might be able to order them directly. I think you mentioned to me privately before that you had plans to do that. So just wondering whether the interaction's already there.
So there's Alan announcing our private features for the future. Yes. So at the moment we have links to Genesis. So if you go to a passport page, if there's links, if it exists to Genesis, we've got those there. But we have been talking to Mattia, and we would like to look at ways that we could allow people to create their groups in Germinate and then pass that off to Genesis for ordering with the gene banks. I'm not sure when that's going to happen,
Okay.
but that's certainly on our list of things that we'd like to be able to do.
Okay. And the second question I had was just, what are the restrictions on this? How big are you planning? Are you planning to, at the moment, is this initially just for the BOLD program? Are you inviting pretty much anyone in the science community to upload their information, and then this becomes somewhat of holistic in terms of data that's been evaluated on breeding pre-breeding lines or crop wild relatives of
So we're absolutely not
gene
inviting.
materials, which could all be only linked, but
Yeah, so we're absolutely not inviting everybody to do it, but I think as part of Bold and this project, like if we have trusted partners who might have data that would add value to what we've got already, then we're absolutely interested in doing that. It's not a free for all for anybody to go and upload data, but I think we need to have those discussions with people who might be able to, yeah, as I say, complement what we've got already or bring value to it.
yeah, sure.
Shiv, you had your hand up.
And I have only question about, because we have been working with the crop test for so many years, and we have a lot of data. When we have submitted data to you, it was only restricted to the recent data which we have taken up with the project. Recently 2 projects we complete, Templeton as well as the one with the Tamari, that one.
Yeah.
With the barley and grass pea were the target crops. So, but before that also we had this crop test project on grass pea. Shall we upload that data also? And so that will help us to have a complete data set available on all the lines of grass pea which have been targeted in different crop trust project. And second things, how can we have a kind of same identity number? Because what happens many times when we have shared the germplasm with the partners, they have given some other numbers, and so are we going to have something like DOI number something, or it is just what numbers I provide that is going to be taken?
So, as you know, Shiv, I'm always advocating that you pick a number that identifies something. As we also know, that doesn't always happen. So we can have synonyms within Germinate, so you can have different names. It's not ideal. It does happen though. So it would be better if we picked something like a DOI. So we've got these things called PUIDs, which everything else has as well. So it's permanent unique identifier. We need to start looking at that really seriously now so we can track things. With regards to the other data, of course, that's going to bring value to this, and we can have that discussion. And if you give us that, then we're more than happy to have a look at that. I don't know if you want to add anything, Sebastian, about unique identifiers or anything, or if that's answered the question.
No, I think you covered it pretty well.
everybody, please pick unique identifiers and stick with it, so we can at least have a trace of where things are and what came before it and where it went. Anybody else got any questions?
Yes, if I may, because I need to leave at 12. I'm sorry. My question would be, what are the prerequisites that you would accept phenotypic data sets? Because they can be very diverse and they can be of good or poor quality. So how would you make a decision that a phenotypic data set enters the database?
Yeah. So in terms of quality, we've left that down to the groups we're working with to decide if they're of high quality or not. So these projects, we're working with individuals. So we've done a lot of work with Shiv, with Alan, lots of other people. It's really, it requires somebody who knows about the data to look at it first and make that call as to whether they're happy with it or not. And we don't do much else with it after that. Our tools allow you to look for things, so you can do like the scatter plots and stuff, and it might identify some issues, but we're generally hoping by the time it gets to us that it's been through some sort of curation phase and it's reasonable. Saying that, we can also keep data sets private within Germinate. So if you submitted us some data, Kirsten, and you said, I'm not 100% sure about this yet, can we use your tools and we can look at it? We can keep that private until you're happy, and then click a switch and it becomes public then, or delete it or make the changes required. So there's different ways of achieving that, but in general, it's up to the person submitting it to determine its quality.
Okay, thanks.
Paul, I have a quick question for Kerstin. Based on this first introduction here, what do you think about Germinate? Is this potentially useful for you, or what else could you advise us here from your first look here?
I mean, it looks like a very comprehensive tool that has already thought about a lot of aspects of data that are meaningful and relevant. And I think it's a useful tool. Of course, now there are many different approaches of establishing databases for plant genetic resources, etc. I think it would be very good to connect the people working on these different database approaches and probably come up with a harmonized approach, or if it's possible to even connect databases with each other, but that's a very complex story, I guess. Yeah,
I think connecting databases is the way forward, because everybody has their own specializations and things. So like us being able to link into Genesis or Eurisco or other tools that might be coming out of some of the nice barley work from IPK. I think it's important that those groups that's their speciality are maintaining those things and providing those links. So there's
yes.
initiatives like, is it FIDARE, part of an EU project about how you start bringing FAIR data together and provide search interfaces onto that to find other people to find things. And we're certainly also involved with like EBI groups in the US as well, about looking at how we make things more searchable, make things easier to find.
Yeah, I think here from IPK it would be good to connect with a group of Uwe Scholz, who have established databases for Bridge and now also for the Agent project, and also with the GenBank documentation group around Stefan Weise, hosting Eurisco, and Markus Oppermann, who's also involved both in the Agent and the Increase project, that also established databases for the large pools of plant genetic resources we are dealing with. So it would be nice maybe to initiate a meeting as well.
Well, we already speak to Uwe.
Okay,
we have grant funding to provide links between IPK and James Hutton Institute. So we've had staff going across a couple of weeks ago, in fact, and we also do a bit of stuff with Stefan Weiss as well through various things, and we're involved in various projects as well. So those discussions have started, which is good.
Okay, that's great, because they are the better persons to comment on this. I'm just a user, but from a user look, it looks already very, very nice, the platform.
The users are the most important people, though, because they're the ones that have to use these things.
True.
technical people have a reputation for just assuming things are obvious to people. And one of the things that we've done with
That's
Germinate,
true.
which
Yeah.
has been quite good, is doing user testing with people. So we've sat down, we've published about this as well, about, can people actually do the things that you want them to be able to do with it, and how we can improve systems to make that better for people. Very, very important, users.
Okay, I have to leave to the other meeting. I'm very sorry, but Salar can update me, or Benjamin can update me on what was said afterwards. I wish you all a nice weekend.
Thank
Bye.
you. See you later.
Thank you.
Okay, are there any other questions?
Hi, do you hear me?
Yeah, go for it, Nadine.
First of all, I would like to thank you, Paul and Sebastian, for the nice presentation. So I have a question. So I think all of us, we have all many data to upload to Germinate. And in our case, in Adicarda, so we have this data in our breeding management system called BMS. So what we need actually when you request data is to take this data from BMS and to put it in the template that you send. If there is if we can develop any new approach to transfer this data directly from BMS to Germinate, because it looks similar with some, of course, with some difference. So this is my comment. So it will be easy and while saving time, because it's the same, I think.
It's similar. It's not the same. In terms of developing tools, yeah, we're happy to speak to you about doing that. That would need to be something that people who worked with BMS did, though. But we'd be happy to work with them to look at ways that we can make that process more fluid and maybe easier on us all. So yeah, absolutely. But until that time happens, we do have templates, and people would be expected to use those templates to put data in. But let us follow up with you, Nuruddin, about that, because I do agree that there's probably things that we could do that makes it easier on all of us. Yeah.
It will be very grateful for us and for you also to.
Yeah. Daniel, you have your hand up.
Yes, thank you for the meeting. And at least I've had a brief look through Germinate, much as I came through a little late. I feel it looks more comprehensive than what I've been previously been more familiar with. But maybe I'll transfer the challenges that I faced briefly with the BMS straight to Germinate and see if it is something you people have already catered for. One has been, I've been dealing with the importation of germ plasm sometimes, but then where we have always had a lot of issues is duplication of entries. Let me say maybe the genotypes. You realize with the other system, if you write something, say maybe starting with the capital or starting with the lowercase, it will identify it as something different. Yeah. So then I was wondering, is there maybe like a format of naming whereby we can use so that we avoid such errors? So how do we cushion against duplication of the same genotypes in a system? Thank you.
So I'll quickly say something, and Sebastian might want to talk about some tools he's been working on to look for potential duplicates. But that's an issue. We didn't allow people to upload their own data for a long time until recently, because you always have things like that, and you say to people, "Are there any duplicates?" No, there's no duplicates. It's all fine. I've checked it. It's never the case. I mean, we're all the same. These things are sometimes difficult to spot. Sebastian's been working on an interesting tool that allows you to paste your names in, and it does. What, Sebastian, do you want to mention that?
Yeah, it's like it's a two-part thing. So first of all, Germinate has got functionality to merge duplicates. So if you once you identify the duplicates, you can select them and tell Germinate to merge them into one entry. Now to find the duplicates, we've got another tool, which, as Paul was saying, you paste the names into it, and then it'll run an algorithm across them to try and identify possible duplicates, and it'll give you a breakdown of the things that it thinks, the items that it thinks are exact matches, and the ones that are potential duplicates, and then you can make the decision whether they actually are duplicates. And that output that that tool generates, you can drop that into Germinate to then merge the entries in the database.
Does that answer your question?
Yes, thank you.
Peter?
I mean, a bit of a follow-on from that one is the related germplasm. So perhaps some material that's a line drawn from one of your original accessions. And then you've got some phenotype data. I was wondering if you can just say a little bit about how you relate the germplasm together as you go through a breeding program with the generations and back to the original passport data.
So I think we probably need to do more on that, but we do have like pedigrees and stuff just now, which will allow you to show some sort of hierarchical relationship between bits of germplasm within a database.
Yeah.
But it's certainly, and I'm well aware that there's more we need to do around it.
I'm aware that there's a field that allows you to associate your original accession, well, your new germplasm, which might be a self line, with the parent. Using this is not totally straightforward. I'm wondering whether there's scope for some more training on how to actually make sure everything is oriented correctly as it's entered.
Yeah, okay. I'll take a note of that. We can certainly. I mean, I'm going to come on to that, something like that in a second. But like any ideas of stuff that people are not sure about that could benefit from additional input from us, then we're more than happy to do that. Surendra, you got your hand up.
Yes, Paul. Thank you. Thank you very much for the nice presentation. And just I would like to mention, I impressed the selection of the data sets on the basis of values. That will be helpful for any breeder to identify their breeding programs, to which trait we have to identify, and it is very easy. By the tool of Germinate, we can identify the sum of the selected lines in a whole group. Otherwise, we have to search single by single. So I very impressed for this tool by the Germinate one. So thank you very much for the developing such kind of application or tools. Thank you.
Thank you.
Thanks.
Anybody else with questions? If not, I have a couple of final slides if I can just show those, and then we can maybe finish a bit early. So it's always good if a meeting finishes early. 2 secs. I'll get this. Okay, so thanks everybody. We'll send out a copy of these slides to people who we have details for, and if you've provided your email address when you logged into WebEx, we should have your details and we'll send that out. But if not, please share them more widely as well. It would be fantastic if you can do that. We've also been recording this, and we can make this recording available to people as well to have a look at again if you want to. There's also a selection of online training we've developed for Germinate, and the link's here, but we'll send this out so you can have a look at it. And it basically goes through each of those sections that Sebastian talked about, but will allow you, it'll talk you through it, and it'll give you some questions that makes you basically use the system. And it'll tell you the answers and stuff as well. It's not a test for everybody, but it'll be quite useful if you're thinking of using it to give you a better idea of the sorts of features it's got. The other thing is, we'd like to get some feedback on this. So this is the first time we've done this, and we want to make it as good and as useful as we possibly can. So again, when you get an email after this meeting with links, there'll be a link to this Google Forms, and we'd ask that if you could just take 2 minutes of your time to sit in and provide honest answers on how we can make this training better in the future. Finally, we're happy to answer any questions. We've got an email address, germinate@basic.uk. Please contact us at any time. You can also get us on social media, on Twitter at the moment. With all the stuff that's happening on Twitter, I'm not sure how long that'll be, but contact us on Twitter, and also links to more Germinate resources. I'd also like to point you in a direction. There's been a couple of Germinate papers. So there's one in 2017 and one in 2021. Links here. Encourage you to go and have a look through those. The second one in particular involves a lot of people that have been on this call, and it's related to the Crop Trust Cropwood Relatives project that just finished earlier this year. Finally, all this work would not have been possible without a whole variety of funders that we have. In particular, this project was funded by the Templeton World Charity Foundation and the Crop Trust, but involved partners like Shiv, who was here at CARDA, Damaris at ICRISAT, and a whole pile of other different funders who have funded Germinate over the years that we'd like to give thanks to. And finally, thank you. We've done our questions bit, but if there's any other questions, you can shout out now. If not, we will be in touch with the resources. And thank you very much. Ben, you've got.
Yeah, thank you very much again, and really indeed for this excellent training. But please remember these Germinate databases. They are being developed for you, for the community. So please use them and provide feedback to Paul and Sebastian. And maybe there will be always expert users, and then you can train your colleagues. Yeah, please use your instances, your databases. Oh, thank you.
And give us feedback, because the more people that ask for something, the greater chance there is that will happen. And a lot of people ask for similar things. So it's well worthwhile. If there's anything that you see that we can't do that you'd like to do, just get in contact with us, and we can have that discussion. Good. Well, thanks everybody. Again, thanks for coming along. Thanks for taking an hour and a bit out of your days. I hope you enjoy the rest of your day. Some of you will be off to bed, I guess. Alan will be getting late. Some of us here in Scotland are just starting our days. But thanks everybody, and we'll be in touch soon.
Bye. Thanks everyone.
Thank you.
Bye.
Thank you, Paul. Thank you, Sebastian.
Thank you very much. Bye bye.
See you later.
Bye. Thank you.
Bye.
Bye.
Bye bye.